time series models for modeling brain signals. So some of you have actually heard a portion of this talk before, and this is ongoing work with Hernando Ombao, who is at UC Irvine, and he's actually going to be here, I believe, the last week of this workshop for the biological part of this workshop. Sure, okay, great. So neuroscience experiments, the type of data that we're looking at. So I don't know if you've ever been a subject of an experiment before. So I had the pleasure of being part of an fMRI experiment. Stuck me in a scanner for about an hour, visual stimulus response. Now you're in there, I was in there for about an hour, and after a time, it got really boring. Same thing over and over and over again. The reason why we have to do this is because brain processes, the signal is very small. There's so much noise in there. So by presenting the stimulus over and over and over again, then their logic is that we're going to average out over the entire course of the experiment. That way we can cancel out all the noise and obtain the signal. So that's their logic. So we have poor signal to noise ratio. Now, if we're looking at, say, EEG or MEG signals, then what you're going to have is you're going to have a lot of characteristics of non-stationarity. So one very simple example of this is if you have, say EEG data of someone who goes through an epileptic seizure. So prior to the seizure, fairly stationary, then boom, seizure hits, and you have high frequency oscillations that come into play. So that's something that we would need to consider. And something that's very unique to this work is the fact that like I was saying earlier, that we have many replicates of the time series data. So what I'm going to talk about here is that the data are actually not identical, so therefore averaging over the entire experiment is, may not be appropriate. So, and I call this kind of like how we have in locally stationary time series models where the second order properties are changing over time. Here we have that property as well as it's also changing over the course of the experiment. So let me give you a very simple example of how this works. Okay? Suppose I have an experiment with, say, 100 trials, 100 replications. So here's trial number 1, trial number 50, trial number 100. Bivariate time series, we look at the first dimension, and we see something that's low frequency oscillation evolving to high frequency oscillation, and the other way around for the second dimension. We see the exact same things happening for replica 50 and replica 100, and so on and so forth. So I made this data up, so this is a toy example, and so if you look at this, you might even say, like, hey, okay, they look almost exactly the same. Let's average over the course of the experiment. But by looking at this data, here's what you cannot see. If I look at evolutionary coherence between the two, the correlation between the two signals over frequency band, in the first replicate, coherence is essentially 0 within that replicate. And halfway through the experiment, we have weak correlation that's slowly dying to 0. And in the in the end of the experiment, we have something that's much stronger and dies down to 0 much faster. So if I were to look at all the coherences over time in the experiment, so that's represented by all these black lines. But what a lot of what's being reported in a lot of the neuroscience experiments is not the black lines, but rather the average, this red line here. So that's what they're reporting. They're going to average over the course of the entire experiment. Now, what I propose to do instead is report something else, and this is what I call a time replicate time plot. So x-axis here is time. You read the same way. So the beginning of a trial is at the left-sided axis, and the end of the trial is on the right-sided axis. And then the y-axis here is what I call the replicate time. So beginning of the experiment is in the top of the plot, end of the experiment is at the bottom of the plot. So what corresponds to essentially a flat line here, 0 coherence, is you can see it's dark blue here. And towards the end, it's much stronger, dies down to 0 faster, which you can see here in the bottom of the plot. But what you can see with this plot, though, is how it's slowly changing over the course of the experiment as you read down the y-axis. So this is what I'm proposing that we report. It's more complete description, a more accurate description of the phenomenon at hand, as opposed to just reporting the average. Now, sometimes reporting the average will be is fine as it is, but this is a more informative plot. So this work was motivated by a particular experiment. So let's go straight to the data analysis, actually. So this is a learning association experiment performed on a monkey. So we collected local field potentials from a monkey, It's a very invasive experiment, and it's not really done a lot because we drilled right through the skull of the monkey and placed electrodes in 2 regions of the brain, the hippocampus and the nucleus accumbens. And the way the experiment works is as follows. Monkey's sitting there, joystick in hand. He's fixating on a point. We present him a picture. Then we present 4 doors. And what the monkey's supposed to do is, given the particular picture, he's supposed to choose one of the 4 doors. So if he were to say, saw a picture of a flower, like here, he's supposed to pick, say, the door to the left. If he saw a house, he picks the door to the top, so on and so forth. So, and then we give him feedback. If he picked the correct door, then we show him a little video of him. It's like a virtual navigation, and we show him a green door afterwards, indicating that that was the correct door. Otherwise, we present a red door, telling him that it's an incorrect choice. And the key here is that the monkey doesn't know the association between the door and the picture and the door. He's supposed to learn it over the course of the experiment. So when he does get it correct, we feed him juice. That's the reward. Otherwise, no juice, red door only. That's it. So, kind of give you an idea what the data looks like. So this is from one trial. I apologize. This actually should have been a dot. And that fixation dot is supposed to be in all of them. But so this is our bivariate time series, and you can see clearly some non-stationary characteristics. For example, the hippocampus. Oh, we have this low frequency oscillation all of a sudden present after the picture is on, and so on and so forth. Even though that the trial is actually much longer, including the video and all that navigation stuff, we're only interested in the first 2 seconds because this is when he makes a decision between seeing the picture and making the choice on what door to pick. And so these are the only segments of the trial that we're interested in. So 2 seconds worth of data, and we yield roughly 2,000 time points here. So, yep. Oh, sorry. This is nucleus accumbens and hippocampus. These abbreviations. Yep. So now to show you the results of this, Hernando and I, we were interested in coherence between these 2 signals, the evolutionary coherence at certain frequency bands. So these are your time replicate time plots that I talked about earlier. Again, x-axis is time within a trial. Y-axis is over the course of the experiment, beginning of the experiment, end of the experiment. So what we see here in particular is the alpha band here. So we see weak correlation slowly increasing towards the end of the experiment. And the other ones, draw whatever conclusion you want. So I did derive some asymptotic properties here and how to do confidence bands here, point-wise confidence intervals. And so looking at the alpha band alone, this is the surface, and then lower bound is the blue, upper bound is the red. The time axis is here. So beginning of trial, end of trial, middle to the right. The replicate time axis is read from left to middle. And if you look at the lower bound alone, you can see it slowly increasing. So, the significance level is becoming more and more pronounced towards the end of the experiment. However, I do want to point out that these are point-wise. It's done at every single point. So this isn't really a band of sorts. It really should have been much wider. But that's something that we're still working on. So this is for coherence. We can also look at power spectrum on each one. So this is a power spectrum, I believe, for the nucleus accumbens. So the takeaway here is theta bands driving the process, the slow waves, and nothing really going on with other bands. And for the hippocampus, exact same thing. And then we're seeing these spikes when the picture was presented. So what we learned here from a scientific perspective was actually nothing new. They already knew that the low frequency bands from 1 to 8 hertz is driving the processes here, even in the learning experiment. But what has not been seen before, because of the lack of existing models and methods, is when you have replicated time series, how to account for the fact that you have non-identical time series going on. And so that's what this model is doing. So to kind of give you details, just a brief overview of how the model works. First, let's look at weak stationarity. So we've seen the Cromera representation yesterday a lot. So our bivariate time series is written by oscillation, sine and cosine waves, oscillating frequency omega, amplitude and correlation being driven by transfer function matrix A of W, and then orthogonal increment process DZ. So This was extended by Rainer Dahlhaus. He talked about it yesterday as well for the locally stationary time series, where now the transfer function matrix is a sequence, doubly indexed sequence. And there are also other variations to this, so I'm just presenting his. And to just have a natural extension of his work, this is what I end up using. So now we have 4 indices here. We have time and replicate time. So where R here is the number of replicates in the experiment, and little r is where you are in the course of the experiment. Now, one of the keys, so the way to estimate this, I did this in a 2-stage fashion. In the first stage, just treat every trial as independent. So then you have an entire literature out there on locally stationary time series on how to estimate the spectrum per replicate. So I'm just going to do it one replicate at a time. So use your, what I'm doing was just your moving your smooth periodograms, localizing time. There's other ways out there, the wavelet periodograms that we talked about yesterday, or selects Hernando's work, Wenchen goes smoothing spline de novo, so on and so forth. You have an entire literature out there. So you do that per replicate, then conditional on your results there will be the second stage where we do smoothing over time and replicate time. So I treat it as a surface estimation problem after that. So I'm not going to go into details here. But, I'm going to go to a little bit more extensions of this work. One thing that I was not fond of here is that there is a lot of smoothing going on. So in particular, first stage, smoothing over frequencies. I was using smooth periodograms. Second stage, smoothing over time and replicate times. And John Aston actually pointed this out to me when I presented this a year and a half ago in Japan, that a lot of smoothing, is there anything that you can do better? And which inspired me to work on this further with Hernando. So moreover, I actually only took a subset of the experiment. The experiment's actually a little bit larger. So let me give you some details on this. So here are 2 plots. This is the performance, whether or not the monkey got it correct or incorrect. So that's represented by the y-axis. It's just a cumulative probability. And here in the x-axis is the trial number. The top plot is for when he had to learn the association between door and picture and door. Bottom plot, he was already trained. He's familiar with it. So he made a few mistakes in the very beginning, but then he ended up getting it correct, and he leveled out to getting nearly 3/4 correct towards the end of the experiment. And whereas in the where he had to learn, so this is where the learning was happening, but he only ended up getting about half the novel trials correct. So he's still making mistakes even towards the end. But it is, if he were guessing by chance, because there are 4 doors, random chance will be 25%. So he is getting somewhat better than random chance. And of course, for the trials that the associations that he was familiar with, he's getting just 3/4 of those trials correct. So this is a type of, I guess, how to quantify if he's learning or not. Now, I also want to show you the experimental design here. There's a lot going on in this picture. First, let's look at the shades of gray here. Light gray, dark gray. This it's a block design experiment. So the light gray, those were the trials when he was familiar with it, familiar with the association between picture and door. Dark gray is when he had to learn. So we have, so it's a block trial, block design, about 50 trials of familiar, then about 200 novel trials, novel associations, then switching back and forth, so on and so forth. Now, the y-axis here is the log periodogram. I average it within the low frequencies, the delta band, and I did that per trial, and that's what the blue represents. The red line here is just a moving average. I just moved the blue lines. So this is what I meant earlier, that maybe the smoothness assumptions over replicate time, or in this case over the trials, is not good, because you can see these blue that may not actually be that smooth. Moreover, you can see here towards the end that's a whole lot more volatile. One thing that this clearly indicates, so that the trials are definitely not identical, so you definitely should not be averaging over the whole thing. So that's what we're trying to account for. This is what we see here, and you might also see that there are these blue lines, that some of them are missing. We actually do have some missing data problem going on here. That's because sometimes when we presented a picture, the monkey responded too fast, or there were just he just made some mistakes with the apparatus, and so those trials were excluded from the experiment. So there's a whole lot more problems with this data set than what was presented earlier. So this is for the hippocampus, for nucleus accumbens. We see a very similar picture here. Again, if we just look at the trend here in the red line, that we're seeing the decrease in the power in the low frequencies. And so how can we relax the assumption of smoothness over frequency and over replicate time? So Hernando and I, we've been pursuing a different approach to modeling this. So let's just back up to something very simple here. Oh, and sorry, I should also bring this up, that this is actually a pretty important question from a scientific perspective. So if you were presented with familiar trials versus novel trials, is there any difference between the two, at least in the power spectrum? So how can we answer this question? Because at the end of the day, this is something that the scientists are interested in. Okay, so this is the model that we're still developing. First, let's start with something very simple. So Rob Crafty, in a biometrical paper published a few years, very recently, he used the following locally stationary time series model. So this is an extension of Wenchen Go's model. It looks similar to the dollhouse model, but here there's not a sequence over a doubly indexed sequence. J here represents subject. K represents measurement of a subject. So subjects were being measured multiple times. And so what he developed here was a mixed effects spectral analysis. And so he's able to account for within subject correlation if a subject were measured multiple times. So that's what he has here. Now, this is for weakly stationarity. If we go to local stationarity, Li Chin and Wenchen Go, along with Crafty's, backing up to the weak stationarity, given this formulation, we can model the log periodogram with a bias correction, with gamma here being the Euler-Mascheroni constant. So here we have the mean over all subjects, subject-specific deviation, and then your error, which has mean 0 and variance pi squared over 6. So this is the model that he used in that biometrica paper. Now, for local stationarity, we have this model here. So now your transfer function matrix is changing over time. Again, this is more of a following Wenchen Go's local stationarity model. And here VJ represents covariates, let's say, if you have covariates in your experiment. And so, so subject J, so you have a time series for subject J, and so you have subject-specific spectra, and that's changing over time as well. So given this model formulation, you get something very similar, but now your mean structure is also changing over time. So this could be block K in your within a trial, let's say, or within a subject. So otherwise, it's the exact same thing. Bias-corrected log periodogram, changing over frequencies and time, and so is the mean function and the subject-specific deviations from the mean. So given all that, I'm doing something very similar to what they're doing, but now I don't have subjects. I just have one monkey. However, I have many trials for that one monkey, the trials being denoted by R. So here, replicate-specific spectra. So given this model, this modeling framework, we can define the replicate-specific spectral density, time-varying spectral density matrix, and therefore also form a model for the log periodogram. So this is your bias-corrected periodogram for some frequency or frequency band theta at replicate R. Then you have the mean for that. So across the entire experiment, replicate-specific deviations, and then any error associated with it. So now to estimate all this, we turn this into a non-parametric regression problem. So we do basis expansions here for both mu and also for delta. The one thing that's difficult here is if we want to account for correlation within across all the trials. One thing that Hernando and I are currently investigating is the coefficients for the replicate-specific deviations. We're modeling that with an AR1. So we're trying to keep it as simple as possible. Now, I brought up earlier the mixed effect spectral model that Rob Crafty developed. We cannot use that here because in many frameworks you have independent units, in that case subjects. Here we have no independent units because we only have one monkey and all the trials are correlated, so we cannot use, at least to my knowledge, some of the existing literature cannot be used here. So this is the modeling framework that we are currently pursuing. We can also extend this to just essentially it becomes just nothing more than a linear model, where for novel trials we have its own mean structure with replica-specific deviations, and same thing for familiar trials. This is also, I do have some preliminary results, but unfortunately I think it's a little too preliminary to show you right now. Hernando and I are still fine-tuning some of the algorithms for estimating these parameters here. To conclude here, I just want to kind of do a little advertisement. So one thing that's really coming up for in the past few months is that the rise of neuroscience experiments and therefore the need for statisticians. So Paul Alivisatos from UC Berkeley is calling the studying of the brain to be an important challenge. And because of that, we have seen in this past April, President Obama announcing the BRAIN Initiative, and also the Human Brain Project conducted here in Europe. So there's the Human Brain Project is based in Geneva, Switzerland, and I do remember seeing like job postings over there. They need people who can do time series, and since this is somewhat of a time series crowd, I'm kind of just advertising it, because there's not a lot of statisticians out there with time series expertise pursuing this kind of work. So there's a lot of things here that may be of interest to you. For example, if we're looking at EEG, MEG, or in my case, I would look at LFP data set. You have something you have a very rich data set in terms of time, and now if you want to look at fMRI, you also have space. So so if you're interested in, say, spatio-temporal models, then this is it for you. Now, there's also a lot of problems that, well, maybe problems, but it could be interesting for you regarding the dimensionality aspect that we have to deal with. So, example, in fMRI, we break up the brain into 300,000 different spatial locations. So, modeling the spatial correlation as well as temporal correlation is a very daunting challenge, and it's still very much an unsolved problem. And there's a lot of things going on in this field. And one thing that I've been involved recently is in imaging genetics. So you have thousands of SNPs out there. You want to associate that with hundreds of thousands of things in the brain. So you want to talk about massive data set. This is what Tom Nichols called big data squared. So big data in terms of space, big data in terms of the genes. So there's a lot of things going on here. And if you've never been involved with analyzing neuroscience data sets, fortunately, coming out soon is a book co-edited by Hernando, Martin Lindquist, Wes Thompson, and John Aston. So this is kind of an introductory in some sense. You'll get one thing that's very important when it comes to analyzing this kind of data set is knowing the science behind what it is. That's not just a vector x of t, that you'll also get like what the data means. And so there's going to be a little bit of that. I'm also involved. I'll be contributing to this book. Ivor Cribben, who's also here, is also contributing. So there's a little bit of us here, and Hernando will be here later during this conference. But it would be, So all I'm saying is that we need more statisticians getting involved, and so I'd like to throw that out there as, to suggest maybe that this is something of interest to you. So I'll end it there, and just would like to thank people who contributed to the data set, Sean Patel and Emad Eskandar from Boston. Thank you.
Could you please show this picture more at the beginning with the different bands, where you had the coherences over the different bands? So what would be the interpretation? So you have on the one hand side, you have rescale time, this is clear, and then you have the replicate time.
Yep, that's y-axis.
And you said on one slide that you average over both times, which probably means local average, right? I mean,
What I'm doing, yes. So what currently happens right now is they'll average over the entire y-axis. That's averaging over the experiment.
but then you lose the learning effect. Right?
Exactly. So by
But
not averaging,
this is not in these pictures. They are not smooth over the entire axis, right?
this is smooth, yes. This is smooth over time, replica time.
But locally.
But locally.
Yes.
Sure.
Okay. And one question is, have you thought of getting out directional information?
The direction from.
I mean, this is coherence is somehow a correlation measure,
Yes.
frequency-specific correlation measure, and people. So neuroscientists sometimes have examples where the influence in one direction, say from one variable to the other, you can see in the gamma band, and
Yeah.
for example the feedback loop you can see in a different band, and so sometimes they plot the transfer function. So basically the A in this Cramer representation rescaled, which is heuristically, but basically it's similar to Granger causality,
Yes.
where you have that.
So the directionality here will be, say, from hippocampus to nucleus accumbens, and that correlation feeding from one region to the other, or vice versa. I'm not very fond of such information. My experience with it from fMRI experiments is that it's just, I haven't found it to be a very, the utility of it I can see, directionality, but I think that the type of conclusions that will be drawn from it, when I tell my collaborators this is what it is, then they will end up saying it's causal in a physiological sense, which is not the case. So I try to avoid doing that thing. So in general, I do think that providing such information is useful, but I find it dangerous to be a little bit more than the benefits here.
Just two little technical questions. The first one, why do you choose the log periodogram approach for your model? If I'm not mistaken, you cannot model coherency, coherency or correlation, cross-correlation with this, so I'm a little bit surprised.
Yes, I turned it now into just a univariate problem.
Yeah, so it's for the autospectra, basically.
Yes,
or-
that's right.
Okay.
That's right.
And the other question is, I'm a little bit, I fully understand the philosophy behind this time evolution over replicates. This is a nice approach. I'm a little bit surprised that nobody in literature did something more probabilistic, like instead of taking the mean, looking at a histogram or another, summary of the data where you could kind of, investigate a little bit more closely what's going on over replicates. Is it true that nobody did that?
One variation I've seen in the context of fMRI, say like a learning type of experiment, is that they will upweight or downweight your estimates, but they will have to use an auto-sample data set. So based on this, that I know that, say, it is that they're still learning, maybe we should downweight or upweight or whatever. But to do it within one model, I have not seen it. I have not seen it at all.
One last question.
My question is simple. When you do your experimental design, you separate your novel and the femoral.
Yes.
Why your block, your femoral, always gets short points times time than the? Because when you mention your question, you want to compare whether there's any
Yeah.
statistical power between these two.
Yeah,
But
that's
when you
a very good point.
experiment, you
What's the
got-
question? I didn't hear.
So the number of familiar trials is significantly smaller than number of novel trials. That's a very good point. Unfortunately, I cannot answer that question, why it was designed in such a manner.
Because when you try to get some idea about how to test the statistical power between these two, you have to design your experiment
Yeah.
properly,
Yeah.
the control and the other things. This is quite important in medical things.
I agree completely, but I don't have the answer to that question.
Okay, let's end that there. Thanks, Mark.